talk about DNA, the role DNA plays in the transfer of genetic material from offspring to progeny across all species. Uh, we will find out how DNA was discovered as the genetic material. In the beginning, proteins and DNA both competed for that. DNA was discovered in 1869. However, it took about pretty much the first half of the 1900s to conclusively demonstrate that it was DNA that was passed down the generations, and that codes for information, codes for proteins, which perform most of the structural functions in specialized cells. So here we have the structure of DNA. As you can see, a twisted helix, a double helix. This is a space-filling model here shown. The bases are in the middle here, the base pairing between A, T, and CG. Four bases in DNA, and we will have another structure later. But here we are showing a double helix. This is what was discovered as one of the major findings. And another scientist named Chargaff, he showed that the composition of adenine was equal to thymine, and the composition of guanine was equal to cytosine across all species. So here we are showing a double helix. This is DNA. So the question is, how did the model, or how the models of DNA changed over time as new evidence emerged? As we said in the beginning, it was either proteins or DNA. We will see evidence from these experiments and relate them to the features of DNA. So let's look at some of the discoveries that resulted in understanding the DNA model. DNA was known since 1869, was thought to store phosphorus. Until the early 1900s, it was thought that proteins had heritable genetic information, not DNA, because proteins have about 20 monomers and DNA have only 4. So scientists thought DNA was too simple to store information. At the most, there could be 64 possible combinations from 4 nucleotides. And obviously, with 20 amino acids, there could be a lot more. So just people thought DNA was just not complex enough. It was too simple, just 4 monomers. So this whole idea, this slew of ideas grew from 1900s to about 1944. Swiss scientist Miescher separated nuclei from the cytoplasm and found nuclein that had phosphorus, not sulfur. At that time, many techniques were coming. Mass spectrometry could determine the molecular weight of a compound. We could also do experiments through which we could find out the composition of a compound, elemental composition, because electron microscopy had been or was being developed. And when all this happened, we knew it was a 1-1-1 ratio of bases, because the percent composition couldn't be figured out until Chargaff. 1928, Griffith experiment showed that bacteria passed DNA when they infected mice. And we'll talk about that in a little bit here. I'm giving you the chronology. After that, even Chargaff, around 1940, he showed the 1-to-1 ratio of DNA bases is incorrect, and the percent is different. In 1944, the famous Avery-McLeod experiment that showed in viruses DNA was genetic material, and then came the very famous Hershey-Chase experiment. So here in 1928, Griffith shows how a transforming element could change one strain to another. Before that, it was thought that bacterial strains couldn't change from one type to another. And this whole experiment basically gave us this idea of transformation, which happens through phyli, or where genetic material DNA can be transferred from one bacteria to another. This is how drug resistance works against antibiotics. So this experiment showed that there was a transforming element. Griffith took two strains of bacteria. There's an R strain, S strain. So S strain is the one that causes disease, is pathogenic. R strain doesn't. But what happens if you take R strain, inject it into mice, the mice will survive. You take S strain, inject it in the mice, they do not survive. But what he did was he took the pathogenic S strain, killed it with heat, so the sugar coat is gone. The only thing that was left was DNA. And so he put that heat-killed S strain, mixed it with living R strain, and then injected mice with that. So mice were killed. So the question was, how did the R strain, living R strains, when they were mixed with heat-killed S strain, how did they kill the mice? So this basically led to the idea that something changed the R strain into pathogenic S strain. So they were basically taking the genetic material from that remnant of the killed S strain and then making that coat, and then became pathogenic and killed the mice. So this was one evidence. The next one, in 1944, Avery and McLeod showed that DNA was the transforming element. So basically, heat-killed S strain can still pass on DNA, right? So they use controls, DNAs, proteases, RNAs. What happens during transformation of R strain? So this is showing how DNA you can separate actually from strawberry or animal cells all look like this, like a strain here in the bottom, precipitating using ethanol and other solvents. So this is the Avery experiment. The rough strain and the smooth strain here, heat killed smooth strain, and then rough strain and heat killed smooth strain. In these two cases, the mice die. As you can see, heat killed smooth strain by itself doesn't kill, but heat killed smooth strain, if you mix it with rough strain, that kills the mice. Smooth strain kills the mice, yes, because it's deadly. Rough strain doesn't. So when you heat kill the smooth strain, the capsule is gone, they're not pathogenic, and so they don't kill the mice. However, when you mix that heat kill strain with rough strain bacteria, they kill the mice. So it shows us here that there is something going on into those rough strains and making them pathogenic. In 1952, a landmark experiment by Hershey and Chase using bacteriophage, which are viruses that inject DNA in bacteria, using radioactivity. Basically this was like a big confirmation about this whole thing. So they took these viruses, grew them with radioactive sulfur and radioactive phosphorus, correct? So the sulfur went to the phage proteins. The sulfur is present in protein and phosphorus is present in DNA. So these phages were then used to infect the bacteria. Phage proteins were separated from bacterial cells through centrifugation. So the bacterial cells formed a pellet in the bottom. In the case of radioactive sulfur, where it was used, the radioactive material was not passed to the next generation. But when these phosphorus labeled phages were used, the radioactivity stayed in the bacterial cells and moved on to the next generation. This basically showed that DNA was the hereditary material. Here is a picture of a phage. This has a tail, and this is how it's injecting DNA into the bacterial cell. And this is the schematic of the experiment. So you have DNA, you have a phage, you have a bacterial cell, This one has radioactive protein. In this case, you have phages with radioactive phosphorus. So here's a blender that is used, and then to separate the proteins outside the bacteria from the bacterial cells. And in the pellets, you will have the bacterial DNA. After centrifugation, in this case, where the radioactive sulfur was used, the radioactivity stayed in the liquid where the protein was. But in this case, the radioactivity was present in the pellet where the bacterial cells were present. This showed here that it's DNA that is present in this. Now, the next round here, as it shows, and that actually was the next round, Rosalind Franklin, a scientist in England, King's College, London, used the concept of Maurice Wilkins, who was a very well-rounded scientist. He also worked on the atomic bomb. So he was thinking about using X-ray crystallography to find the structure of substances. Rosalind Franklin used this X-rays. Basically, she took some DNA fiber. There are two forms of DNA. One has more water than the other, A versus B form. So she used the B form and passed X-ray through it and got a very nice picture, which was used by the famous scientists James Watson and Francis Crick to figure out the double helix. The irony here is that I'll show you. So this is the diagram that was shown to Watson without Rosalind Franklin's permission. In this case here, the cross shows a double helix. There are 10 bases on top of each other in each turn of the helix, which is like 3.4 nanometer. And the distance from one base to the other is obviously one base of like 0.34 nanometer times 10. And one base is missing here because of stacking, repeating bases, and the sugar and phosphate will be on the outside. This was the experiment where Rosalind Franklin is using X-rays on DNA fiber, and here's the image on an X-ray film. So let's go back here. What we are saying is all of the three male scientists got the Nobel Prize in 1962. Rosalind Franklin died in 1958. She could have received it. So basically, some people say that this work was stolen by the three men, and the lone woman did not get the credit, which she should have. And that has happened throughout history. It still happens. And this rule where posthumously you cannot be awarded the Nobel Prize was made only in 1974, so she could have received it. And then here we have the molecular basis of inheritance. This is the structure of a DNA strand. Sugars and phosphate, backbone, they're on the outside. In the middle, here are the bases, and there's a long linear sequence, and the two go in opposite directions. That would be anti-parallel. And here is the numbering. So the numbering would be, as you can, in this case, the two chains are twisted around each other. The bases are in the middle, stacked. Sugar phosphate outside. Here is the schematic, chemical structure. A binds T, C binds G, and it's a 1-2. Basically, the amount of adenine equals the amount of thymine, and amount of guanine equals the amount of cytosine.
15 minutes.
15, isn't it? Yeah. So parallel versus anti-parallel. You see the 5 prime I labeled here. 1, 2, 3, 4, 5. So on this end, the 5 is on the top, 3 is on the bottom. On this end, the 5 is on the bottom, 3 is on the top. That's anti-parallel. This is a space filling model that we use. And so basically, this whole thing here shows you how the phosphorus and the carbon, the sugar are outside. Most of the nitrogens are blue in the middle. Oxygen, hydrogen outside here. The bases are right in the center. So this is how the double helix was deduced, and how do you go from genes to proteins? There are it's a tetra 4 nucleotides. It's a triplet. Three nucleotides code for one amino acid. So this is called the codon, and specific sequences. For example, AGT would always code for serine. There's some redundancy, and there will be more as we go along in this course, you will see there are 64 codons, and there's only like 20 amino acids. So there's more than enough to code for the 20 amino acids. And codon is a 3-nucleotide sequence that will specify a particular amino acid during protein synthesis from mRNA on ribosomes. So, to recapitulate, we have shown the different experiments that showed that DNA was the genetic material. We also talked about the work of Rosalind Franklin, who was instrumental behind that discovery, and she should have received the Nobel Prize, which she didn't. And now we will go through some questions. If you have any questions, you can ask. Thank you.